Plants exhibit tremendous diversity in structure and form. Humans use differences in morphological traits such as the size and shapes of flowers, fruits, and leaves, or the overall appearance of a plant to differentiate one species from another. Although they can appear quite different on the outside, plants show a fundamental similarity in basic features of the organs that make up the plant body, the organization of tissues into organs, and the features of cells that make up those tissues. This video explains the basic features of plant anatomy that underlie the diversity of form and function observed in plants and describes a simple technique to observe the different types of cells found in the herbaceous tissues of some common fruits and vegetables. The vascular plant body is composed of two organ systems. The below-ground root system anchors the plant and functions to take up water and nutrients from the soil. Above ground, the shoot system is composed of the stem that forms the main axis of the plant body, leaves that harvest light to make sugars, and reproductive structures such as flowers. A stalk-like structure, the petiole, attaches the leaf blade to the stem at the node. A dormant axillary bud is found at the node. The space between consecutive nodes is called the internode. The basic types of plant cells can be observed in the cross-section of this corn stem. Large, thin-walled parenchyma cells comprise most of the ground tissue that makes up the bulk of the herbaceous plant body. Parenchyma cells have irregularly thickened cell walls and provide flexible support to the stem and petioles. Sclerenchyma cells, such as the sclerenchyma fibers surrounding this vascular bundle, are thickened with lignin and provide rigid support to the plant body. Sclerenchyma can also be in the form of sclerids, also called stone cells, that form the grit that is characteristic of pears. The vascular bundle contains the water-conducting xylem cells and sugar-transporting phloem cells that make up the vascular tissue. Notice how the vascular bundles are scattered throughout the stem of a typical monocot, but the vascular bundles are arranged in a ring in the stem of a dicot. Closer examination of the monocot and dicot vascular bundles show a distinct difference in the arrangement of the xylem and phloem cells in the vascular bundles. Monocot and dicot roots also show differences in organization of vascular tissue. In the monocot root, xylem is arranged in a ring around the central vascular cylinder with clusters of phloem in between. In dicots, xylem cells show different arrangements, such as the X pattern shown in the center of this root, with phloem cells in between the arms of the X. Parenchyma, parenchyma, sclerenchyma, xylem, and phloem can likewise be found in the leaves of vascular plants. Woody tissues are composed of millions of xylem cells and associated fibers. Differences in cells give the different species wood its characteristic grain, color, and strength. Cells of the herbaceous plant body can be observed in many common foods. To view them, prepare a homogenate of the plant tissue by placing a small piece of fruit or vegetable in a blender with a small amount of water and blend for 10 to 20 seconds until there are no large chunks of tissue remaining. Place small amounts of the tissue into microcentrifuge tubes. Label the tubes with the name of the plant and the type of stain to be applied to it. Next, add 2 to 4 drops of a single stain to a tube. Here, toluidine blue is being added to the homogenate. Mix the tissue and stain with a toothpick and place on a rack and allow the stain to color the cells for 10 to 15 minutes. Continue adding stain to each of your tubes until all of your tissues have been stained with the different stains that you will be using. After your tissues have stained, remove the material from your microcentrifuge tube with a spatula or toothpick and place on a microscope slide. Spread the tissue out and then place a cover slip on top of it. Gently press on the cover slip to flatten and spread out the tissue. If there's excess stain on the slide, you can apply a small amount of water at the edge of the cover slip and then tap the edge of the slide onto a paper towel to wick away excess water and stain. Once this is done, your slide is ready to view. Here you can see parenchyma cells from celery that have been stained with fast green. Fast green also highlights strands of parenchyma cells from a celery stalk. Safranin highlights the red sclerids from pears. Safranin also highlights xylem tissues shown here. Toluidine blue stains the parenchyma strands of celery a light purple and the xylem cells stain dark blue. Staining with toluidine blue also makes it possible to see the fine details of the xylem cell walls in broccoli. And last, staining with iodine makes it possible to see the starch grains inside of the common potato.